for Jesse going on board here. We just compared that to the prokaryotic cell and just make sure you know the main organelles and how they work, what their functions are, especially when you go in from the nucleus, so the nucleolus out to the membrane, into the endoplasmatic reticulum and the ribosomes, and know how that works and how they produce proteins that, and we're going to talk about protein synthesis and how that actually works at the ribosomes in more detail later. And then you'll probably go over this one more time and it'll be a little bit easier to understand. But we kind of finished around the fungi, the kingdom of the fungi, and I think we'll just go there and maybe go through the first few slides a little bit faster because we already done those, and then spend some time with their reproductive systems or modes, because they're important in classifying them and understanding the differences between various fungi. And then we'll cover algae, which we won't do too much because really they're not a big concern to us. I mean, they have huge functions in nature, mainly producing oxygen, and they're photosynthetic, so in the ocean they have a huge role there for life, for ecosystems, but they're not that important to us as microorganisms. And then protozoa, which are the unicellular microorganisms, and there's 4 kinds, so we'll get through those, and then parasitic worms. And I was looking, they have worms up there, but they don't have anything the size we're looking at. And why I was looking is actually we talked about the microorganisms being small and too small to see with the naked eye, but that's not really true for some of the parasitic worms. Actually, I don't know if anybody saw it. I was looking for it yesterday, just what's new, and you can look it up. But there was it was really funny because there was this Australian rock star, I guess a punk rock guy, and you could just see the dreadlocks to the side, and it's all shaved on his head. And apparently he had one of those parasitic worms under his head, basically, and he had these seizures, and they thought it could be maybe a brain tumor or something, and then that's what they found. And the thing is, he's vegetarian, and it was a tapeworm, I think, that you usually find in pigs, and he said, how is that ever possible? Like, he was upset about that. And I think it was yesterday in the news, and I think what they said is he ate a vegetarian burrito. So you guys can figure out how that works. But mainly what they think, and I don't even know how they would relate that to anything. I mean, he could have eaten that. I don't know how they did the research, but apparently the chef, and that's what you do, or why they fall under the category, because a lot of times you use the microscope to look at eggs, things like that, to look at reproductive stages. So apparently, those are microscopic, and you can transfer animals like that, parasites like that, through just dirty hands, and so that's apparently what happened. But yeah, they performed a 3-hour surgery and took it out completely. So I think he's not going to have any more seizures. So we'll cover those at the end, and then we'll start with the viruses, and see how far we get there. They're even a step below, so those are the smallest, really the smallest living things, which we file them under, although they don't have the cell structure. And so we'll look at those and just their main structure, how they work, how they become infectious, and how they enter cells, how they leave cells, and how they change basically the host to their advantage, which is really amazing what they can do. So now I gotta jump a lot of slides. I don't know why I'm holding this against the screen. I'll just start with the. So this is what we're gonna do today, partly, and then fungi, algae, protozoa, and parasitic worms, and then viruses. We'll start and see how far we get with those. So we said there are a lot of species, and there's macroscopic ones. The ones we know from just what we consume, what we see, the fruiting bodies of some of those, and then there's a lot of microscopic ones, the molds, the yeasts, and they're usually, the majority is unicellular, but you see them you have some that are have some cellular specialization and multicellular. The basic shapes are those ones, we talked about that too, the old round oval egg-shaped, and this is kind of like a one that's budding, so maybe this one, and then the hyphae, which are long filamentous fungi and molds. Some have both, but that's really the two structures you should relate to fungi. They're all heterotrophic. They do not produce. They do not use sunlight to produce matter, to gain energy from sunlight. They mostly are harmless, and they live off dead plants and animals. So they're very important in decomposing dead matter. They're very important to decomposing dead animals and to make those nutrients again available for new growing organisms. Some of them are parasites. None of them are obligate parasites, so that they're not, like say a virus will not be able to live outside of a host, so that would be an obligate, I guess, parasite in that case. And then we have mycoses, fungal infections, if that, for example, would apply to humans. They inhabit a lot of habitats, and the yeasts, I talked a little bit about their importance to us, but that's the basic shape. We said they're oval, egg-shaped, so that's the basic shape. You find all the organelles we talked about in the eukaryotic cell in here, and they reproduce through an asexual process called budding. So they basically produce, it's kind of like a mitosis, where you basically have a cell division, so you do have all the necessary organelles in a cell nucleus in that bud, but it's a smaller portion, and it basically comes off of the main cell. And if they hang together, if they build a chain, they call them pseudo hyphae or hypha, which is not, they're not really hyphae, and that's why they're pseudo hyphae, because they're really a chain of budding yeast cells, not hyphae. That's the other cellular structure you find, and so that's the one. That one is considered a hyphae, and what's important is if this cellular structure can either be divided, the cells can be divided by a septum with pores, or they don't have a septa, and there's those two kinds, and these really build the mycelium. And the mycelium is massive usually. Like a lot of times you would see fruiting bodies, so you would see a mushroom, and there's one example I'll show you in a little bit. The mycelium can be very, very large in comparison to what you actually see of the fungus. And they're usually cottony, velvety texture, hairy. So you've seen probably yesterday on your plate that stayed on the table some of those mycelia or a mycelium, just filamentous fungi and growing in there. And then a lot of what you usually find is you have a vegetative hyphae that digests, that absorbs nutrients. So it's feeding the fungus. It's the phase that grows and lives, and then you have reproductive hyphae, and they produce spores. And those spores, we said, are nothing like the endospores of prokaryotic cells. So those spores are really there for reproduction, not to make sure that the organism can survive through conditions that are not favorable for it. So those spores are important for reproduction, and there's very many different spores. We'll see a few of those. So just keep in mind there are for reproduction, and there are spores produced asexually, which just means you have one parent organism and it produces spores to create more offspring that grows and uses up or produces its own mycelium. So it's growth of the organism, but it doesn't mean there's any kind of genetic change in that. So there's the sexual reproduction, and we also you'll also see spores. They're just is a different way, and we'll go through how those different ways of reproducing in fungi work. Yeah.
For the long filamentous fungi, when you have septa, or they said they're single cells divided by the septa. What about the ones without the septa, the non-septa ones? And I still see multiple nuclei.
Yeah, so they're cells with multiple nuclei.
So it's one cell with
Yeah,
multiple nuclei.
yeah.
Okay.
So then, as I said, you have a fruiting body on top. In some cases, not always. It's just to show you that this is not really, and we have a slide on that so you understand what you actually see. But if you see a mushroom, in this case, the ones that we really easily identify as a mushroom, you have a fruiting body and you have a mycelium. So this is really the vegetative phase, and that's the reproductive phase. But it doesn't always look like this. It could look many other ways. So and you'll see one probably right here. So again, spores is how they reproduce, and those spores are formed through budding or mitosis, but they come from one parent organism, and that's why it's an asexual reproduction. So if you would, for example, look at something like this, you have a vegetative hyphae, and it just grows. It's got submerged hyphae and surface hyphae. So you have if you have, say, a certain medium, it could be soil, it could be a slice of bread. You would see growth on top, and it usually grows into the medium. And then eventually, when the conditions are right, which a lot of times for fungi means moist conditions, they create reproductive hyphae. And in this case, they look like this, and these structures, which we are getting into in a little bit, then release spores at some point, and these spores actually, fall back or get absorbed by the medium, and then these spores produce germ tubes. So they grow and then slowly turn into hypha or hyphae, and then these hyphae again produce mycelium, and then mycelium again can have vegetative hyphae or reproductive hyphae. So that would be an asexual way of reproducing, because this one plant is just producing a reproductive hyphae if the conditions are right, releasing spores that are growing into a new hyphae. And so this is a picture of one of them. You can see that there's a lot of vegetative hyphae here, and then you got a reproductive one here. So just keep those, and I put this on a couple different slides with not too much information, because we use or scientists use the reproductive mode and the reproductive structures to classify and identify fungi. And so the spores, which you've already seen now in the spore forming structures, are really what you're looking for. So I mentioned before there's asexual spore formation and there's also sexual reproduction, so sexual spore formation. In the asexual reproduction cycle, it just means, and I pointed it out too, you have a single parent cell, and through mitotic division, so mitosis meaning you just have a cell division and you pass on the same genetic material to the next cell. You don't have any kind of change in the genetics. So in general, the next generation has identical genetic material, and that is usually in fungi done by or the reproductive structures that we find in fungi are sporangiospores and conidia. So those are two different ones, two different ways of asexually reproducing.
What's the difference between those two, sporangiospores?
Okay, yeah, we'll get there in a second. Though you can see it, it's probably easier than just me talking about them. So just before we get there, the sexual spore formation, and I kind of hinted to this a couple times now. What's the advantage? I mean, if you have an organism that can asexually reproduce, why would you even consider sexual? Why is it important to have sexual reproduction? And I pointed it out. I mean, you've probably all come across it in some biology class. You know, if you have two parents, you create a gene combination which is different from, which creates a different set of traits or characteristics for the new organism, and so that is different from either parent, and that means this organism may be better adapted to an environment or not as adapted to an environment, and so through that process of maybe being less well adapted to environment and not surviving or not reproducing, kind of lose those traits, and then maybe eventually you have a offspring with better traits, better characteristics, maybe characteristics that allow that organism to move into an area where they have more nutrients available that is not used up by a lot of other organisms. That organism is going to be more successful in growing, also reproducing, and so in the end you have have an adaptation, or organisms adapt well, and that means which is part of evolution. So the more successful, the better adapted organisms are gonna be able to survive and reproduce better. And so you have this process of evolution that's going on over millions of years, and it helped adapt organisms to all kinds of different environments. So before we go into the asexual one and look into the the two different types, you'll also see for sexual reproductions the spores that are formed following fusion of two different strains. So sexual, two different strains, and it's different than in other plants or animals, but we'll go through those. There's the zygospores, there's a ascospores, and then there are basidiospores. But I'll get to those, so I'll lead them on real quick, and then we'll cover each one of those. And just make sure, because it's a little bit confusing, even in the book, I think they didn't do a very good, especially basidiospores, I don't think they really covered that well, because it's really difficult to even see, which to me is essential. If it's sexual reproduction, you have to have two different strains, and if you look at the picture in your book for the basidiospores, you can't really tell. It's like on the bottom you see a little bit, but I put in a different picture so you get a better idea, at least in my how that works. So for the asexual spores, you have sporangiospores. We already mentioned those, and conidia. And really the main difference is the spores here are enclosed in a spore-bearing sac. So you have a sporangiospore, and they're developed through successive cleavages. So you produce a lot of those asexually. So there's a lot of mitotic divisions that produce these sporangiospores, and they're enclosed in this sac called sporangium. And then when this sac opens up or ruptures, you have a release of sporangiospores, and they're able to grow in a medium and then produce probably initially hyphae, vegetative hyphae, and then those can produce or can turn into structures like this again if you have for reproduction. So you have these reproductive hyphae. And then conidia, I don't think I need you to remember all these different kinds. It's just so you know, and you've seen them before. There's like say arthrospores, chlamydiospores. They're different. They're really differentiated by their shape. So these ones are more like cylindrical, round. There's these ones here that where you have multiple. So they call them macroconidia, where you have multiple conidiae, I guess, in this. But you also have micro, depending on the state of the conidia, and then you have porospores over there. But I heard, and that's why I was keeping them in too, or wanting to mention them, besides the fact that you should know that there's asexual spores, sporangio, and conidia. I think there was a discussion also yesterday on the radio about it's the California state prison system, I think, has a lot of problems with a disease called valley fever, and that's I don't know much about it. I just looked into it a little bit, but it is a fungus, or it's a fungal disease, and I think there are some death, and they're trying to really figure out what to do and how to move them, etc. And I think they're caused by a fungus with arthrospores, and those are airborne spores, and so I don't know why the conditions in prisons are such that you see that more than we would, for example. I don't hear much about valley fever in the general population, but it seems like they're airborne, so they breathe them in, and then they can, I think they attach to to the lungs or in the lungs to cells, and so you kind of get coughs and problems with breathing, and I don't know what it does from there, but I think it can cause it can go all the way to infected people dying. So that's just because I heard it on the radio, but it is happening. So just keep in mind that fungi aren't always on our mind when it comes to microorganisms, but they're definitely ubiquitous, just like the bacteria, and you saw that. So they're everywhere, and you saw that in the plates that we just kept outside with the lid off, and there was fungal growth in some of them. So that covers the asexual spores. We're not going to go too much into any other detail because we still have to cover the sexual spores. So remember, we had the 3 different kinds: zygospores, ascospores, and basidiospores. And really, I don't think there's a huge difference. Basically, what you see for all of them, and I put a 1 there just so I know where to start in this picture, but also you know where to start in this picture. But basically, you always have a positive strain and a negative strain, or a female or a male hyphae growing, and they're merging, they're growing together, and because they're 2 different organisms or 2 different individuals, it's sexual reproduction. And so you have the asexual phase, which we've talked about, where you just have spores and they germinate. You have the mycelium, you have a fungal growth, but then you have also a sexual reproductive phase, and that happens when you have this positive strain and a negative strain merge and produce a zygote, and that matures. So this would just be part of this huge underground mycelium. Yes?
Are they actually different, or is it
Yeah,
just for ease of...
I don't know much about their, it's hard to say what their exact anatomy is, or morphology, how they, I don't know how many of these hyphae, for example, if you have 2 different fungi or 2 organisms growing, if there are many of those. I assume there are, but I don't know if they're the positive and negative strain has to be morphologically and probably physiologically different. So they are different from each other, and that's how they're able to merge and produce a zygote. But I don't know what the exact differences are for those. Just remember, in general, just remember that for sexual reproduction, we have those different strains. And so then you have the production of a zygote that matures, and then because it came from 2, there's a step where through meiosis, which means you go from a diploid to a haploid organism, and we can talk about that some other time too if that's not clear. It's not really necessary for now. All it is you have a zygospore, which then releases spores. And so the spores, the organism that grows out of this spore has characteristics, just like we have characteristics from 2 parents, they have characteristics from 2 parental fungi or parental organisms. And so that's the first of 3 sexual reproductive the first of 3 different ways fungi reproduce sexually. This is the second one, ascospores, and there's some more prominent members of that family that I'll show you a couple pictures of. But again, here you have what they call an ascogonium, which is the female one, and then you have an antheridium, which is the male one. So you see that these hyphae here, or in this case positive, negative, you have these hyphae and they have a lot of vegetative cells, but then you also have the reproductive ones, and those are different. So in this case, they fuse just like in the first case, and produce these ascogenous hyphae, and then they basically produce these asci, is what they're called these fruiting bodies, and release ascospores. And they're, again, after meiosis, they're haploid. That means those spores, those ascospores, produce a regular new fungus with genetic material from both parents. And so again, just maybe focus on, you have you have a female and a male hypha, and they're fusing to produce this structure, this sexual reproductive structure, and then they release spores, which in this case are called ascospores. And then lastly, and that's the one where I have a different picture than I had. I changed this a little bit ago, so I don't know if everybody has the updated version of this. If you are using, my PDFs, but I just put this in because it seems a little bit simpler. It's or at least what they point out, and if you look in your book, if you want to, you can see that the basidiospores, I mean the mating types, the positive and the negative, aren't really clear to me. I mean it shows up somewhere at the bottom, but here you really see you have you have the negative and the positive mating type again. That's how you start. I think that's kind of how they end in their picture. So just remember, there's always a positive, negative, female or male, and they produce this dikaryotic mycelium. So they really what you should remember just is that they produce this mycelium, and that eventually produces these what we usually see in our fruiting bodies. So this is really the mycelium in terms of protection of the new generation, and in terms of spreading a new generation. They're producing these fruiting bodies, and that's what we see. And in those, if you ever now see a fungus, you walk around, you find one, look under the cap there and look for the gills, and they're usually lined with basidia. So if you look at a gill underneath here, this is what you see, and these basidia are holding the basidiospores, but not, depending on what stage they're in. They go from basidia to, and then again meiosis to a basidium with 4 appendages, and those release the basidiospores. So you basically have this process in this fruiting body that produces these basidiospores. That's really all you need to remember about them. But there again, it's a sexual form of sexual reproduction where a negative and a positive mating type come together, and then out of that fusion, they create a fruiting body which holds these produces these basidiospores that are then released and germinate into a mycelium until it's time to sexually reproduce again. So those are the 3. I have them. Here they are. So those are the 3 sexual reproductive ways for in the kingdom of the fungi. There's a couple more. Usually they're based on the type of sexual reproduction. There's a couple more. So we did look at the Zygomycota, Ascomycota, and Basidiomycota, and then there's one I think that can be the Chytridomycota. They have flagellated spores, so they're different from these. There's only, I think, one of them is really or is of importance in terms of disease or as a microbe for humans, and then you have a whole group of fungi that fall under the category imperfect because they only have asexual spores. So there's no sexual reproduction in those. So they don't belong in any of these three because they're only reproducing asexually, and the only other exception is this phylum with flagellated spores. So you have Zygomycota, Ascomycota, and Basidiomycota. Those are the three main ones, distinguished from each other by their sexual reproductive cycle. And so the Zygomycota here, for example, you can see one of those structures, and you've got the sporangium with the spores in there. And then this is Ascomycota, and Aspergillus. I don't know if you've heard that one before. Is a pretty important one for the Ascomycota, and Penicillium is the other one. And then this one, under the Basidiomycota, for example, this one is, I think, one that they pointed out in the book in Oregon, and this is a tree stump. So if you see, like, that's the mass, and they claim that the underground mycelium covers about 2,200 acres for this area. So you can imagine how little we see on the surface and how much is going on the ground, how much they're growing underground, and that then is the fruiting body that is able to lead to sexually reproduced spores, but really the vegetative part is underground and much bigger. So when we try to, or when researchers are trying to identify fungi, they usually, you can usually isolate them on specific media. We won't do that here, but that's just like we do it with microorganisms here. And then you do, again, just like we do here for a lot of the bacterial cultures we've worked with, they look at macroscopic and microscopic features of these fungi, and we talked about those. So they look at asexual spore-forming structures, conidia, sporangiospores. They look at the type of hyphae. Are there septa or are there not septa? So that's how they distinguish between hyphae producing or hyphae cell type fungi. Then the texture and the pigmentation of the colony. I mean, yesterday I think we had some. I think I saw a pretty green, dark green fungus in one of those. So, somebody working with fungi a lot might be able to say, well, there's only a certain amount of possibilities if you look at airborne spores. And so in a room like this, I would imagine if it's green, looks a certain way, grows a certain way, this is what we're dealing with. And then the physiological characteristics, always important, just like the genetic makeup. So physiologically means always, what are they growing on? What's their substrate? What do they change the media through their waste, or are they able to live in under certain conditions? I think, for example, that fungus that causes valley fever is supposed to be living in desert regions, so it probably is I don't know what phase of it could be that some of the spores can survive that, but usually you would imagine you need more moisture conditions. So just those physiological characteristics are important to distinguish between them. And then the genetic makeup, that's again more and more important. You know, what are their how does the genetic makeup compare between groups? How related are they? And then what we should have probably started with is what are the roles? You know, what do we know about them, or what do they mean to us? And we looked at, you know, maybe the athlete's foot picture, and you have mycoses, you have fungal infections, and I said that, I think we talked about it last time a little bit, how those can be, those infections can usually be suppressed by the immune system in a sufficient way most of the time. Fungal infections aren't really anything we have to worry about too much, but then, you know, valley fever, for example, in the state prisons is a big issue right now. You see that in immunosuppressed patients a lot, where the immune system is not able to respond adequately to fungal infections, and so that can become lethal in for example, cancer patients. To suppress fungal growth is really an important part of that, or to control that is an important part of the treatment. They're causing allergies, they can produce toxins, and another interesting number is that they think that whatever fruit crop is produced, I don't know if that's worldwide, I would say in the U.S., in like Western countries, but I think that's from the U.S. I had to check on that. They actually consume up to 40%. So it means if we have a certain harvest, almost half of it goes to a kingdom that we're not really ready to share with, and so that's their role.
Just the fruit.
Yeah, I'm sure that I don't know how I'd have to really look up how they determine that for what country, but you know, you would definitely see that for a lot of vegetables, grain products. I mean, anything. I mean, they're really able to grow on most things. I think we pointed out last time that there's almost no organic matter that they wouldn't be able to consume. So you would find that maybe fruit is the one that's most susceptible to it. That could be part of it. And maybe the way we pack and transport other crops is not as easy for fungi to grow on or grow in, but it definitely has a huge impact. And then the beneficial impact really are, I mean, there's there wouldn't be the cycle of life and the decomposing of dead plants and animals wouldn't be possible. I mean, basically any ecosystem would collapse quickly without fungi decomposing all these materials. I mean, you see them probably on, you know, fallen trees in usually more moist forests. You know, they're and what you really see is usually only little what's actually growing in terms of the mycelium underground, etc. We use them in making foods. Saccharomyces are, you know, for beer production, for example, and in other, I mean, there's lots of yeasts. We're using different yeast strains are used for beer production, alcohol production, bread, those kind of things. And then they're also a source of antibiotics. You know, penicillium we saw earlier. There's alcohol, organic acids, and vitamins. So they have benefits be, and especially for us, I mean, you know, it's hard to, it's always, the adverse impacts and the beneficial impacts are really always just what, out of the mind of a human, you know, it's not that the fungi care what we like and what we don't like, but that's just so you know what we're dealing with. You want to take a break before the protest, so we have a mental break before we go into the next group? Good, me too.